The processes of digestion and absorption occur in a liquid environment. Within the digestive system, various glands produce and release secretions that create an aqueous environment, facilitating these processes. In previous lessons, we studied how gastrointestinal function is controlled, as well as the motility of different regions. In this lesson, we will explore the various secretions of the digestive system. When food is taken in and chewed, the first secretion it encounters is saliva, produced by the salivary glands in response to stimulation from the parasympathetic nervous system. Salivary glands consist of a duct system that ends in acinar structures. The acinar cells produce saliva, which is secreted into the lumen of the acinus. Initially, this secretion contains water, mucus, electrolytes, enzymes, and other components. It then travels through the duct system, where sodium and chloride are reabsorbed, primarily under the influence of aldosterone. This process is similar to what occurs in the kidneys. Saliva serves multiple functions. It has antimicrobial properties due to antibodies and lysozymes. In omnivores like rats and pigs, and also in birds, salivary amylase initiates starch digestion. In ruminants, saliva contains a high concentration of bicarbonate, which helps neutralize the acids produced in the rumen. An adult cow can secrete between 100 to 200 liters of saliva daily. In the mouth, food is formed into a bolus to facilitate swallowing. Upon reaching the stomach, this bolus mixes with gastric juice. Depending on the species, we can observe two distinct types of gastric mucosa: fully glandular mucosa in carnivores, and in equines, both glandular and non-glandular mucosa, separated by a line called the gastric fold margin. The glandular mucosa contains numerous crypts that are continuous with the gastric glands. Within these glands, there are several cell types, but I will focus on three: surface mucosal cells, chief cells, and parietal cells. Surface mucosal cells line the surface of the stomach and the crypts. Their function is to produce thick mucus that protects the stomach lining from hydrochloric acid and mechanical damage. This is why anti-inflammatory medications must be administered cautiously. They can reduce mucus production, exposing the mucosa to acid and mechanical injury. Chief cells are responsible for secreting pepsinogen, which is activated into pepsin. Pepsinogen is the inactive form and is stored in granules within the chief cells. When pepsinogen is secreted into the gastric lumen, it is exposed to a low pH, leading to its cleavage and activation into pepsin. Although often referred to as an enzyme, pepsin actually represents a family of enzymes that digest proteins. Enzymes synthesized in an inactive form and later activated in the gastrointestinal tract are known as zymogens. This protective mechanism prevents them from digesting the cells that produce them. Parietal cells are responsible for producing hydrochloric acid. Chloride and hydrogen ions are secreted into the lumen to form HCl. Inside the parietal cell, water and carbon dioxide combine to form carbonic acid, facilitated by the enzyme carbonic anhydrase, which is abundant in the gastric mucosa. Carbonic acid dissociates into H+ and bicarbonate ions. The H+ ions are secreted into the lumen via a proton pump in exchange for potassium, which requires energy. The potassium accumulated in the cell is then secreted back into the lumen alongside chloride. Thus, chloride and hydrogen ions are secreted to form HCl. The bicarbonate produced is released into the bloodstream in exchange for chloride, increasing the availability of chloride inside the cell to produce more HCl. This bicarbonate raises blood pH, a phenomenon known as alkaline tide or postprandial alkalosis. Later, this bicarbonate is utilized by the pancreas to neutralize gastric secretions in the duodenum, which we will discuss later. There are several stimuli for the secretion of hydrochloric acid. When the animal is expecting to receive food, simply seeing the food or sensing its smell initiates the cephalic phase of gastric secretion. During the cephalic phase, the food has not yet reached the stomach, but the mere expectation of receiving food causes the stomach to begin preparing for its arrival. The parietal cell can be stimulated by gastrin, acetylcholine, and histamine. The parasympathetic nervous system stimulates the enteric nervous system to release acetylcholine near G cells and parietal cells. Both cell types have receptors for acetylcholine, so the parietal cell is stimulated to secrete hydrochloric acid, and the G cell responds by secreting gastrin. Gastrin travels through the bloodstream and reaches the parietal cell, and the combined action of both gastrin and acetylcholine on the parietal cell further increases the secretion of HCl. All of this occurs before the food reaches the stomach. When food enters the stomach, the gastric phase begins, amplifying the effects of the cephalic phase. The distension of the stomach wall by the food bolus is detected by mechanoreceptors, which directly stimulate the enteric nervous system to release acetylcholine, acting on both G cells and parietal cells. Additionally, food increases the stomach's pH, which is detected by chemoreceptors and provides positive feedback for hydrochloric acid secretion by stimulating G cells to release more gastrin. There are also enterochromaffin cells that secrete histamine, stimulated by acetylcholine and gastrin, further enhancing hydrochloric acid production. When all three receptors are activated, secretion is maximized. As hydrochloric acid secretion increases, the stomach's pH decreases. When pH drops to around 2, gastrin secretion is suppressed, and it stops entirely at a pH of 1. With gastrin stimulation removed, hydrochloric acid secretion decreases. The duodenum can also influence secretion. When acidic content enters the duodenum, secretin is released, which reduces hydrochloric acid secretion. After food is processed in the stomach, it moves to the duodenum and is referred to as chyme. Here, it is acted upon by pancreatic secretions. The pancreas is a mixed gland, anatomically divided into two lobes, the right and left. The left lobe follows the greater curvature of the stomach, while the right extends along the descending portion of the duodenum. The glandular tissue consists of two distinct parts, one with endocrine functions and the other with exocrine functions. The endocrine portion contains the pancreatic islets, or islets of Langerhans, responsible for secreting insulin and glucagon. The exocrine portion, which makes up the majority of the tissue, is responsible for producing pancreatic juice. After production, this exocrine content drains into a duct system leading to the duodenum. This pancreatic duct system has an important anatomical difference between dogs and cats that is clinically relevant. In dogs, the accessory pancreatic duct is the main one, draining into the minor duodenal papilla. The bile duct drains separately into the major duodenal papilla. In cats, the pancreatic duct joins the bile duct and both empty together into the major duodenal papilla. This anatomical difference predisposes cats to a condition known as feline triaditis, but we won't cover that in this lesson. The exocrine pancreas is composed of interconnected acini through a branched duct system. Acinar cells produce enzymes stored in granules as zymogens or inactive forms. When stimulated, they secrete a fluid containing these enzymes into the acinar lumen. At the junction of the duct and a sinus, centroacinar cells modify the fluid composition by adding bicarbonate, making pancreatic juice alkaline. These cells have receptors for acetylcholine, cholecystokinin, and secretin. Acetylcholine is released by the enteric nervous system, while cholecystokinin is released in response to fats and proteins in the chyme, primarily stimulating acinar cells. Secretin, triggered by low pH, mainly stimulates centroacinar cells to secrete bicarbonate. Pancreatic stimulation occurs during the cephalic and gastric phases of digestion and intensifies during the intestinal phase when stomach contents enter the duodenum. The liver is an organ with numerous functions, one of which is acting as a gland in the digestive system that secretes bile. It exhibits significant anatomical variation among species. Externally, it is divided into lobes separated by fissures. Microscopically, it is composed of lobules. Each lobule forms a hexagonal structure made up of plates of hepatocytes interspersed with sinusoidal capillaries that direct blood toward the central vein. Hepatocytes also form bile canaliculi connecting to the bile ducts. Bile consists of water, cholesterol, electrolytes, bilirubin, and bile acids. One of the primary components of bile is bile acids, synthesized from cholesterol. These molecules possess both hydrophilic and hydrophobic portions, giving bile its detergent-like action, which aids in fat digestion by making lipids soluble in water. Hepatocytes synthesize and secrete bile into the bile canaliculi, which flow into bile ducts and are stored in the gallbladder. It's important to note that horses do not have a gallbladder, so bile drips directly into the duodenum. When chyme enters the duodenum, especially if it contains fat, cholecystokinin is secreted, causing relaxation of the sphincter of Oddi and contraction of the gallbladder, directing bile into the duodenum. Bile acids, after facilitating fat digestion, are absorbed in the ileum and returned to the liver for further bile synthesis. This recycling of bile between the liver and intestines is known as enterohepatic circulation. In our next lesson, we will study the processes of digestion